Welcome to MCAT Notes to Fall Asleep To. I'm really glad you're here. Today we're talking about amino acids, and I want to be upfront. This is a topic that most people experience as a chart with 20 rows and 3-letter codes. Maybe if your textbook was feeling generous, you'd even have a color-coded diagram. But I've been thinking about amino acids a lot lately. And the thing that keeps striking me is how much story is buried in that chart. Like, here's something that's been on my mind. Every amino acid your body uses to build proteins is left-handed. Every single one. And not just yours. Every bacterium, every plant, every animal on Earth uses the same handedness. The mirror image versions are perfectly stable molecules. Chemistry doesn't prefer one over the other, but life does, and nobody fully knows why. We're going to get to that, but we're also going to talk about why a single amino acid swap causes sickle cell disease, why vitamin C deficiency makes your connective tissue fall apart, why the taste of Parmesan cheese and the biochemistry of falling asleep trace back to the same metabolic pathway, and why one amino acid is the reason newborn screening exists. My goal is that by the end of this, the 20 amino acids won't feel like a table to memorize. They'll feel like characters you actually know. So relax, and let's get into it. Let's start with the basics. What is an amino acid? Picture a carbon atom. In biochemistry, we call this the alpha carbon, and it's the center of everything. Bonded to that carbon are 4 groups. An amino group, that's a nitrogen with 2 hydrogens, NH2. A carboxyl group, a carbon double bonded to an oxygen with a hydroxyl hanging off COOH, a lone hydrogen atom. And then the 4th thing, the side chain. It's also called an R group, which gives each amino acid unique chemical properties. 3 of those 4 groups are the same across all 20 amino acids. The only thing that changes is the R group. And this is where the design gets elegant, because the fact that the backbone is standardized is what allows biology to work. The ribosome, the molecular machine that builds proteins, has one mechanism for building a protein. The peptide bond always forms in the same way. The amino group of one amino acid reacts with the carboxyl group of the next, zipping them together like beads on a string. Once the chain is assembled, the side chains take over. They determine how the protein folds, what it binds to, whether it's an enzyme or a structural scaffold or a channel in a membrane. 20 amino acids, 20 side chains. And from those 20, the human genome builds somewhere around 20,000 to 25,000 different proteins. Though honestly, that number undersells it. With alternative splicing, post-translational modifications, and combinatorial complexity, the actual functional proteome is orders of magnitude larger. Okay. Now I need to tell you about one of the deepest mysteries in all of biology. And it starts with your hands. Hold up your left hand. Now hold up your right. They look the same, right? Same fingers, same structure, same connectivity. But try to perfectly overlay one on the other. You can't. They're mirror images that can't be superimposed. And molecules can be exactly the same way. When a carbon atom is bonded to 4 different groups, it's called a chiral center. The word chiral comes from the Greek word for hand, chir, and because the alpha carbon of an amino acid is bonded to 4 different things, the amino group, the carboxyl group, the hydrogen, and the R group, it's chiral. Can exist in 2 mirror image forms. There's one exception, glycine. Glycine's side chain is just a hydrogen, So its alpha carbon is bonded to two hydrogens, which means only three distinct groups, not four. So of the 20 amino acids, glycine is the only achiral molecule. Every other amino acid, however, can exist in two possible forms, left-handed, which we call L configuration, and right-handed, which we call D configuration. Every chiral amino acid in your body is the L form. Now, chemically, there is no reason to prefer L over D. They have identical stability, same energy, same melting points. If you synthesize amino acids in a flask with no biological input, you get a perfect 50-50 mixture of both forms. It's called a racemic mixture. Nature, on its own, doesn't pick sides, but life did. At some point, billions of years ago, something broke the symmetry. And whatever that event was, it locked in a choice that propagated across every branch of the tree of life, every bacterium, every plant, every animal. L-amino acids. Only. Why? This is one of the great open questions in origin of life research, and there are some genuinely wild hypotheses. One of my favorites involves neutron stars. Neutron stars emit circularly polarized ultraviolet light, and there's experimental evidence that circularly polarized radiation can preferentially destroy one enantiomer over the other. So the idea is that in molecular clouds, the interstellar nurseries where amino acids form on dust grains, this polarized light created a slight excess of L-amino acids. Those amino acids hitched a ride on meteorites. The meteorites fell to early Earth, and that tiny L excess was enough to tip the balance. The evidence is out there in a rock from space. The handedness of your proteins might trace back to the death of a star. Let that sit with you for a second. All right, let me give you the practical tools. There are actually two different naming systems for chirality, and they both show up on the MCAT. The first is the L/D system. You draw a Fisher projection. That's the cross-shaped diagram where horizontal lines point toward you and vertical lines point away. Then you look at the amino group. If it's on the left side, that's L. If it's on the right, that's D. Left equals L. That's the one to burn into memory. The second is the R/S system, which uses Kahn-Ingold Prelog priority rules. Here's the process. Step 1. Identify the four groups on the chiral center. Step 2. Rank them by atomic number of the atom directly bonded to the chiral carbon. Highest atomic number gets highest priority. Hydrogen, with atomic number 1, always gets lowest. Step 3. Orient the molecule so hydrogen points away from you. Step 4. Trace a path from priority 1 to 2 to 3. Counterclockwise? That's S, from the Latin sinister. Clockwise? That's R, from rectus. And here's the elegant part. For amino acids, L and S almost always agree. All L amino acids are S. The only exception is the amino acid cysteine. Cysteine is L but R. And the reason is sulfur. Cysteine's side chain contains a sulfur atom, and sulfur has atomic number 16, higher than oxygen at 8 or nitrogen at 7. So when you apply the CIP rules, the sulfur bumps the side chain's priority up, flipping the direction of the path from counterclockwise to clockwise. Same L configuration on a Fisher projection, but R in the Kahn-Ingold Prelog system. Oh, and one more thing. These chiral amino acids are optically active. They rotate plane polarized light. You can literally shine a beam of polarized light through a solution of L amino acids and watch the plane of polarization twist. It's the three-dimensional geometry of the molecule physically interacting with the electromagnetic wave. Now, let's actually go through the structure of all the different amino acids. And I want to do this by group because the grouping tells you something fundamental about how proteins work. We're starting with the nonpolar, non-aromatic amino acids. These are the hydrophobic ones. They don't play well with water. And in a folded protein, the hydrophobic effect, which is arguably the single most important force in protein folding, drives them to the interior, away from water and into the core of the protein. One thing people don't always appreciate about the hydrophobic effect, it's not that these nonpolar residues are attracted to each other. There's no strong attractive force pulling them together. It's that water molecules prefer to hydrogen bond with each other rather than organize around a nonpolar surface. Burying the hydrophobic residues releases those ordered water molecules, increasing the entropy of the system. The protein folds because it's thermodynamically favorable for water. There are 7 nonpolar, non-aromatic amino acids. The mnemonic is GAVLIMP. Grandma always visits London in May, please. First, glycine. Glycine is the smallest, most flexible amino acid whose side chain is just a hydrogen atom. You can find it in collagen, which is the most abundant protein in your body. It's in your skin, your bones, your tendons, your cartilage, and its structure is a triple helix, 3 polypeptide chains wound around each other in a tight rope. The interior of that triple helix is so cramped that every third residue must be glycine. Anything bigger and the chains can't pack tightly enough. The entire structural integrity of collagen depends on glycine being small. Here's where it gets medically relevant. Osteogenesis imperfecta, sometimes called brittle bone disease, is often caused by mutations that replace one of those glycines in collagen with a larger amino acid. One glycine replaced by, say, an arginine. And the triple helix destabilizes. Bones fracture easily. Connective tissue weakens. The consequences of losing that one tiny hydrogen side chain are devastating. And here's a detail I love. Glycine is also an inhibitory neurotransmitter. In your brainstem and spinal cord, glycine acts on chloride channels to hyperpolarize neurons and reduce excitability. The smallest amino acid in your proteome also helps keep your nervous system quiet. There's something poetic about that. Next, we have alanine. Its side chain is a methyl group CH3. In research, alanine has a very specific role. It's the default substitution for mutagenesis. When biochemists want to figure out whether a particular amino acid's side chain matters, they mutate it to alanine. This is called an alanine scan. Alanine's side chain is so small and so bland that replacing any residue with alanine essentially deletes the original side chain's contribution without introducing a new interaction. If you mutate a critical residue to alanine and the protein loses function, you know that residue was doing something important. But if the protein still works, maybe that residue was just along for the ride. Valine has an isopropyl side chain, 3 carbons branching at the beta carbon. If you look at the structure, it literally looks like the letter V. It's one of the 3 branched chain amino acids, along with leucine and isoleucine. Now, here's a disease connection that every MCAT student needs to know. Sickle cell anemia. The molecular cause of sickle cell disease is a single amino acid substitution in the beta globin chain of hemoglobin. Position 6. The normal residue is glutamate, a negatively charged hydrophilic amino acid. The sickle cell mutation replaces it with valine, a nonpolar hydrophobic amino acid. This one substitution is enough to create a sticky hydrophobic patch on the surface of deoxyhemoglobin that causes the molecules to polymerize into long fibers. Those fibers distort red blood cells into the sickle shape, which causes them to clog small blood vessels, leading to pain crises, organ damage, and shortened life expectancy. Leucine has 4 carbons in the side chain. Arranged as an isobutyl group, leucine is the most abundant amino acid in proteins. If you picked a random residue from a random protein, your best bet would be leucine, and it's typically used as a signaling molecule. Leucine is a potent activator of mTOR, the mechanistic target of rapamycin, which is the master regulator of cell growth and protein synthesis. When leucine levels rise in a cell, mTOR gets activated and tells the translational machinery, nutrients are here, start building. This is why leucine gets special attention in exercise science. After resistance training, muscle protein synthesis increases, and leucine is one of the key signals driving that response. The BCAA supplement industry is built on this biochemistry. Whether supplementation beyond normal dietary protein actually helps is still debated. But the signaling pathway is real and well characterized. Next is isoleucine, and the name itself tells you what it is, an isomer of leucine. And this means you have the same molecular formula, just a different arrangement. An isoleucine has a structural feature the MCAT loves: 2 chiral centers, the alpha carbon, like all amino acids, and the beta carbon, which in isoleucine is bonded to 4 different groups. Among the 20 standard amino acids, only 2 have 2 chiral centers, isoleucine and threonine. And since we're talking about branched chain amino acids, valine, leucine, isoleucine together, let me tell you about maple syrup urine disease. This is when urine and earwax develop a sweet maple syrup-like smell. It's caused by a deficiency in the branched chain alpha keto acid dehydrogenase complex, which is the enzyme that breaks down these 3 amino acids after transamination. Without it, branched chain amino acids and their keto acid derivatives accumulate to neurotoxic levels. It's rare, about 1 in 185,000 births, because it ties together amino acid metabolism, enzyme deficiency, and clinical consequences. Next up, methionine. The side chain is a straight carbon chain with a sulfur atom in the middle. One critical note: methionine contains sulfur but does not form disulfide bonds in the same way that cysteine does. Here's what makes methionine extraordinary. The codon AUG codes for methionine, and AUG is the universal start codon. Every single protein synthesized by your ribosomes begins with methionine. It's the first word of every protein sentence, the opening note of every molecular symphony. Although, to be fair, methionine aminopeptidases often clip it off afterward, so the mature protein might not start with it. But the ribosome always begins there. Methionine is also the precursor to S-adenosylmethionine, or SAM, and SAM is arguably one of the most important small molecules in all of biochemistry. It's the universal methyl donor. DNA methylation, SAM provides the methyl group. Histone methylation, SAM. Neurotransmitter synthesis, SAM. Phospholipid biosynthesis, SAM. One amino acid sitting at the metabolic hub of the entire methylation network. The epigenetic code that controls gene expression runs on a currency that traces back to methionine. And finally, in this group of nonpolar, non-aromatic amino acids is proline. Proline side chain is rather unique in that it loops back and bonds to the nitrogen of its own amino group to form a 5-membered ring. Technically, this makes proline an amino acid rather than an amino acid, though it's always included in the standard 20. And that ring has a profound consequence. It constrains the backbone. While every other amino acid has free rotation around its N to C bond, proline is locked. The Fuanchi angle is fixed at roughly negative 60 degrees. This rigidity means proline can't participate in a regular alpha helix. Alpha helices require specific backbone dihedral angles, and proline can't adopt them. So when a proline appears in a sequence, it introduces a kink, a bend, a disruption. That's why proline is called the helix breaker. Protein structure prediction algorithms flag proline residues immediately, because wherever proline shows up, the secondary structure changes. Proline is also essential in collagen formation. The repeating GlyXY motif of collagen puts proline and hydroxyproline in the X and Y positions. Hydroxyproline is a post-translational modification where vitamin C acts as a cofactor for prolyl hydroxylase, and that's the scurvy story. Without vitamin C, you can't hydroxylate proline. Without hydroxyproline, the collagen triple helix is thermally unstable. It literally melts at body temperature. Your connective tissue falls apart. Gums bleed. Wounds don't heal. Teeth loosen. Sailors in the 18th century solved this empirically. Citrus fruit prevented scurvy. But it took another 200 years before anyone understood that the molecular explanation was a single hydroxylation reaction on a proline residue in collagen. Alrighty, now let's talk about the 3 amino acids with aromatic side chains: tryptophan, phenylalanine, and tyrosine. These side chains contain aromatic rings, planar, conjugated ring systems with delocalized pi electrons. And because of those pi systems, they absorb ultraviolet light. This is actually practically important. If you've ever worked in a biochemistry lab, you've measured protein concentration using UV absorbance at 280 nanometers. That signal comes primarily from tryptophan and tyrosine. So you're literally counting aromatic amino acids to quantify protein. Tryptophan is the largest amino acid. Its side chain is a CH2 group attached to an indole ring, a benzene ring fused to a pyrrole ring, and it's recognizable by its double ring structure. Tryptophan is the biochemical starting point for serotonin. The enzyme tryptophan hydroxylase adds a hydroxyl group to the indole ring, producing 5-hydroxytryptophan. Then aromatic amino acid decarboxylase removes the carboxyl group, and you get serotonin, 5-hydroxytryptamine. Serotonin regulates mood, appetite, sleep, and gut motility. About 95% of your body's serotonin is actually in your gut, not your brain. And then, in the pineal gland, serotonin is converted to melatonin, the sleep hormone. So here's the metabolic arc. An amino acid in your food becomes the molecule that regulates your mood during the day, which then becomes the molecule that helps you fall asleep at night. Tryptophan to serotonin to melatonin. This one singular pathway connects your diet to your emotions, to your circadian rhythm. And yes, I know about the Thanksgiving turkey myth. Turkey does contain tryptophan, but so does chicken, and cheese, and tofu, and pretty much every protein source. What actually makes you drowsy after Thanksgiving dinner is the massive carbohydrate load. Insulin spikes from the carbs clear competing large neutral amino acids from the blood, which helps tryptophan cross the blood-brain barrier more effectively. So it's the mashed potatoes and the stuffing that amplify the tryptophan effect. The turkey is just the scapegoat. Phenylalanine is the next aromatic amino acid. Its side chain is attached to a benzene ring. And phenylalanine is where I want to tell you one of the great public health success stories of the 20th century. Phenylketonuria, PKU. It's a genetic disorder where the enzyme phenylalanine hydroxylase is deficient. Normally, this enzyme converts phenylalanine to tyrosine. Without it, phenylalanine accumulates in the blood and gets diverted to alternative metabolic pathways, producing phenylketones, compounds that spill into the urine. If untreated, the accumulation causes irreversible intellectual disability. But here's the triumph. In the early 1960s, Robert Guthrie developed a simple bacterial inhibition assay that could detect elevated phenylalanine in a drop of newborn blood. That test, called the Guthrie test, became the foundation of newborn screening programs worldwide. Today, in the United States, every single newborn is screened for PKU within 48 hours of birth. If caught early, the treatment is a low phenylalanine diet for life. And with dietary management, cognitive outcomes are essentially normal. It's one of the first examples of precision medicine, decades before the term existed. Identify the genetic defect. Understand the biochemistry. Intervene before the damage occurs. Oh, and here's a detail that ties things together. You know the warning on diet soda cans? Contains phenylalanine. That's because aspartame, the artificial sweetener, is a dipeptide of aspartic acid and phenylalanine. When you digest it, you release free phenylalanine. For most people, that's irrelevant. For someone with PKU, it matters. Notice also what phenylalanine hydroxylase does. It converts phenylalanine to tyrosine. That's not just a degradation pathway. That's a biosynthetic connection. Tyrosine is made from phenylalanine, which means if you have PKU, you're also potentially deficient in tyrosine and everything downstream of it, which is a lot. We'll get there. Next, we have tyrosine. Tyrosine is basically phenylalanine with a hydroxyl group added to the benzene ring, turning it into a phenol. That one OH group changes everything. It makes tyrosine more polar. It makes it capable of hydrogen bonding. And most importantly, it makes the amino acid phosphorylatable. Phosphorylation is the addition of a phosphate group catalyzed by kinases. And tyrosine kinases are some of the most important enzymes in cell biology. When a receptor tyrosine kinase on the cell surface binds its ligand, a growth factor, an insulin molecule, it autophosphorylates its tyrosine residues. That phosphorylation creates docking sites for downstream signaling proteins. And here's why this matters clinically. Many cancers are driven by overactive tyrosine kinases. Mutations that make these kinases constitutively active, stuck in the on position, lead to uncontrolled cell growth, which is why some of the most successful cancer drugs in the last 2 decades are tyrosine kinase inhibitors. Imatinib for chronic myeloid leukemia, erlotinib for lung cancer. These drugs specifically target the aberrant phosphorylation on tyrosine residues. They exist because we understand the chemistry of this one amino acid's hydroxyl group. Tyrosine is also the precursor to dopamine, norepinephrine, and epinephrine. Your entire catecholamine neurotransmitter system traces back to tyrosine. And melanin, the pigment in your skin, hair, and the eyes, and thyroid hormones, T3 and T4, which regulate your metabolism. Tyrosine is the upstream molecule for an astonishing range of biology. Now we cross a line. From hydrophobic to hydrophilic, from the interior of the protein to the surface, the polar amino acids, like water, they form hydrogen bonds. They participate in the aqueous world. We'll start by talking about the uncharged polar amino acids that have polar side chains but don't carry a formal charge at physiological pH. Serine. Serine's side chain is CH2OH. It's basically alanine with a hydroxyl group. That hydroxyl group can be phosphorylated by serine kinases. And here's a number that puts things in perspective. In the human phosphoproteome, roughly 86% of all phosphorylation events occur on serine, about 12% on threonine, and about 2% on tyrosine. Serine is the workhorse. The vast majority of cellular signaling runs through serine phosphorylation. Serine is also at the heart of a class of enzymes called serine proteases. These are enzymes that use a serine residue in their active site to hydrolyze peptide bonds. Trypsin, chymotrypsin, elastase, thrombin. These enzymes use a catalytic triad, typically serine, histidine, and aspartate working together, to perform nucleophilic attack on peptide bonds. It's one of the most elegant examples of catalysis in all of biochemistry. 3 amino acids, arranged in 3-dimensional space with angstrom-level precision, creating a reaction that would otherwise be negligibly slow at body temperature. Next, we have threonine, which looks like serine, but with an extra methyl group. And like isoleucine, threonine has 2 chiral centers, the alpha carbon and the beta carbon. So, those are your 2 amino acids, isoleucine and threonine, that have 2 chiral centers each. Threonine's hydroxyl group can also be phosphorylated, making it the second member of the phosphorylation trio: serine, threonine, and tyrosine. Threonine is also an essential amino acid, which means your body can't synthesize it. You have to get it from food. And it plays important roles in mucin glycoproteins, where the hydroxyl group is a site for O-linked glycosylation. Mucins line your respiratory and gastrointestinal tracts, and their heavy glycosylation creates the slimy, protective layer of mucus. So threonine is helping protect every surface in your body that faces the outside world. Now, let's talk about asparagine and glutamine. I want to talk about these together because they share a key feature, the amide group. Their side chains end in CONH2, a carbonyl bonded to an amino group. That amide nitrogen does not ionize at physiological pH, so it doesn't accept hydrogen atoms. Asparagine and glutamine are polar. They can form hydrogen bonds through that amide, but they stay uncharged. This is different from the basic amino acids, where the nitrogen-containing side chains do get protonated. Asparagine, by the way, was the first amino acid ever isolated. 1806. French chemists Louis-Nicolas Vauquelin and Pierre-Jean Robiquet extracted it from asparagus juice. The name literally comes from asparagus. Glutamine is the most abundant free amino acid in human blood, and it's a major metabolic fuel for rapidly dividing cells, especially immune cells and enterocytes, the cells lining your small intestine. During critical illness or major surgery, plasma glutamine levels drop sharply because demand outstrips supply. There's been significant research into whether glutamine supplementation improves outcomes in ICU patients, though results have been mixed. Glutamine also functions as a nitrogen shuttle. Amino acid catabolism generates ammonia, which is toxic, glutamine synthase attaches that ammonia to glutamate, forming glutamine, which can safely travel through the blood to the liver and kidneys for processing. It's basically a molecular taxi for nitrogen waste. Cysteine is where things get really interesting. Cysteine's side chain has a thiol group, SH. It's like serine, but with sulfur instead of oxygen. And that sulfur gives cysteine a capability that no other amino acid has, the disulfide bond. When two cysteine residues come close together in three-dimensional space, their thiol groups can undergo an oxidation reaction. Each loses a hydrogen, and they form a covalent sulfur-sulfur bond, also known as a disulfide bridge. This is one of the strongest covalent interactions in protein structure, and it's critical for stabilizing extracellular proteins. I emphasize extracellular because the intracellular environment is reducing inside cells glutathione, which itself contains a cysteine residue, keeps the cytoplasm in a reduced state that breaks disulfide bonds. But outside the cell, the environment is oxidizing, so disulfide bonds form and persist. Antibodies, insulin, digestive enzymes, they're held together by disulfide bonds because they operate in oxidizing environments. Here's a tangible example. Your hair. Hair is made of keratin, which is rich in cysteine. The pattern of disulfide bonds between cysteine residues determines whether your hair is straight, wavy, or curly. When you get a perm, the chemicals first reduce those disulfide bonds, break them apart. Then the hair is reshaped. Then new bonds are reformed by oxidation. You are literally doing redox chemistry at the hair salon. And remember our earlier discussion. Cysteine is the 1L amino acid with an R configuration in the Kahn-Ingold prelog system. The sulfur's atomic number flips the priority ranking. Cysteine is the exception to the rule in more ways than one. One more thing. Don't confuse cysteine with cysteine. Cysteine is the free amino acid with SH. Cysteine is two cysteines linked by a disulfide bond, the oxidized form. One letter difference. The MCAT will test it. Now, let's talk about acidic and basic amino acids. There are two amino acids that are negatively charged at physiological pH, aspartate and glutamate on the MCAT. You'll almost always see the deprotonated names, aspartate, not aspartic acid, glutamate, not glutamic acid. Because at pH 7.4, the carboxyl groups in their side chains have already lost their protons. COOH becomes COO minus, carrying a negative charge. In its side chain, aspartate has one methylene group and then the carboxylate. Aspartate has critical roles in metabolism. It's a key player in the urea cycle, where it donates a nitrogen atom that ends up in urea. It's also part of the Molet-aspartate shuttle, which transfers reducing equivalents from the cytoplasm to the mitochondria. And in the catalytic triad of serine proteases, aspartate is one of the three partners. It helps orient the histidine, which activates the serine. Glutamate is just one CH2 group longer than aspartate. Glutamate is truly extraordinary. First, it's the most abundant excitatory neurotransmitter in your brain. When a glutamatergic neuron fires, it releases glutamate into the synapse, where it binds to receptors like NMDA and AMPA, receptors on the postsynaptic neuron. NMDA receptors are particularly interesting. They require both glutamate binding and membrane depolarization to open, which makes them coincidence detectors. They're central to long-term potentiation, which is the molecular basis of learning and memory. Second, Glutamate is the precursor to GABA, gamma aminobutyric acid, which is the brain's primary inhibitory neurotransmitter. The enzyme glutamic acid decarboxylase removes a carboxyl group from glutamate to produce GABA. So the main excitatory neurotransmitter is the direct biochemical parent of the main inhibitory neurotransmitter. Excitation and inhibition linked at the molecular level. Your brain's accelerator pedal is one enzymatic reaction away from its brake pedal. Third, glutamate is the amino acid behind umami. Monosodium glutamate, MSG, is the sodium salt of glutamic acid. Your tongue has specific taste receptors, T1R1/T1R3 heterodimers, that bind glutamate and signal savory, Parmesan cheese, ripe tomatoes, soy sauce, mushrooms, all naturally high in free glutamate. Umami was identified as the fifth basic taste in 1908 by the Japanese chemist Kikunae Ikeda, who isolated glutamate from kombu seaweed broth. Now, let's move on to the three amino acids with positively charged side chains at physiological pH: arginine, lysine, and histidine. Their side chains contain nitrogen atoms that accept protons, gaining a positive charge. Arginine has a long carbon side chain ending in a guanidinium group. 3 nitrogen atoms are in that guanidinium group, and the positive charge is delocalized across all 3 through resonance. This makes the charge extremely stable. Arginine's side chain, pKa, is around 12.5, meaning at physiological pH, it's essentially always protonated and always positive. You'd need a very basic environment to strip that charge away. Arginine also has a beautiful connection to cardiovascular biology. It's the substrate for nitric oxide synthase, which produces nitric oxide, NO. Nitric oxide is a gaseous signaling molecule that relaxes vascular smooth muscle, dilating blood vessels and lowering blood pressure. This discovery was so important that Robert Furchtgott, Louis Ignarro, and Farid Murad won the Nobel Prize in Physiology or Medicine for it in 1998. Next, lysine has a 4-carbon side chain with a terminal amino group. At physiological pH, that terminal amine is protonated, NH3+, positive charge. Lysine's real power is in post-translational modifications. Lysine is the most frequently modified amino acid in histone proteins: acetylation, methylation, ubiquitination, sumoylation. Each modification on a lysine residue changes the interaction between histones and DNA, altering gene expression without changing the DNA sequence itself. Histone acetylation on lysine, for example, neutralizes the positive charge. The acetyl group replaces the proton. Without that positive charge, the histone binds DNA less tightly, the chromatin opens up, and transcription increases. Histone deacetylases, HDACs, reverse this process. Some cancer drugs are HDAC inhibitors designed to reactivate tumor suppressor genes by keeping histones acetylated. So lysine isn't just a structural amino acid, it's a regulatory interface between chemistry and gene expression. And finally, histidine. Histidine's side chain is a CH2 group attached to an imidazole ring, a 5-membered ring with 2 nitrogen atoms. And that imidazole ring has a property that makes histidine unlike any other amino acid. Its pKa is approximately 6.0, which is tantalizingly close to physiological pH of 7.4. This means histidine is the only amino acid whose side chain is significantly protonated and deprotonated at physiological conditions. It exists in a dynamic equilibrium between charged and uncharged states. Every other amino acid side chain is either almost fully charged or almost fully uncharged at pH 7.4. Histidine is the one sitting right on the edge, and that makes it perfect for enzyme catalysis. In an enzyme's active site, where the local pH can shift based on substrate binding and conformational changes, histidine can act as both a proton donor and a proton acceptor. It's a molecular switch that flips at exactly the right moment. The catalytic triad I mentioned earlier, serine, histidine, aspartate, relies on this property. Histidine shuttles a proton from serine to aspartate, activating the serine for nucleophilic attack. Without histidine's tunable pKa, the reaction wouldn't work. And then there's hemoglobin. In the hemoglobin molecule, two histidine residues play starring roles. The proximal histidine directly coordinates the iron atom in the heme group. It's the tether that holds the iron in place. The distal histidine sits on the other side of the heme, stabilizing the bound oxygen molecule and preventing carbon monoxide from binding too tightly. Without these two histidines, hemoglobin couldn't function. Every breath you take depends on two histidine residues being in exactly the right place. And histidine's imidazole ring also participates in the Bohr effect, the phenomenon where hemoglobin's oxygen affinity decreases at lower pH, promoting oxygen release in metabolically active tissues. When pH drops, histidine residues pick up protons, which shifts the hemoglobin conformation from the high affinity R state to the low affinity T state. Again, histidine's pKa, sitting right near physiological pH, makes it the ideal sensor for these small pH changes. I could talk about histidine all day. It's the Swiss Army knife of amino acids. All right, before we wrap up, let's do a quick pass through everything we covered. Every amino acid has the same backbone: alpha carbon bonded to an amino group, a carboxyl group, a hydrogen, and a side chain. The side chain is the only thing that differs. 20 side chains, 20 amino acids. The alpha carbon is a chiral center in every amino acid except glycine. All biological amino acids are L configuration. In the R/S system, all L amino acids are S, except cysteine, which is R because sulfur's atomic number changes the priority ranking. Glycine, smallest, a chiral, most flexible. Alanine, methyl group, simplest chiral amino acid. Valine, isopropyl, branched chain, 3 carbons. Leucine, isobutyl, 4 carbons, most common amino acid in proteins. Isoleucine, isomer of leucine, 4 carbons, 2 chiral centers. Methionine, thiother with sulfur, no disulfide bonds. Start codon amino acid, precursor to SAM. Proline, cyclic, bonds back to its own nitrogen, helix breaker, essential for collagen. All nonpolar, all driven to the hydrophobic core of proteins. Tryptophan, largest amino acid, indole ring, precursor to serotonin and melatonin. Phenylalanine, benzene ring, purely hydrophobic, converted to tyrosine by phenylalanine hydroxylase. Deficiency causes PKU. Tyrosine, phenol ring, that hydroxyl group makes it polar and phosphorylatable by tyrosine kinases, precursor to dopamine, norepinephrine, epinephrine, melanin, and thyroid hormones. All three absorb UV light at 280 nanometers. Serine, hydroxyl side chain, most common target for phosphorylation. Threonine, hydroxyl plus methyl, 2 chiral centers like isoleucine, also phosphorylatable. Asparagine, amide group uncharged because the amide nitrogen doesn't ionize at physiological pH. Glutamine, amide group, most abundant free amino acid in blood, nitrogen shuttle. Cysteine, thiol group, forms disulfide bonds through oxidation, the 1L amino acid with R configuration, the phosphorylation trio, serine, threonine, tyrosine, the 3 molecular switches of signal transduction, aspartate and glutamate, both negatively charged at physiological pH, side chain carboxyl groups deprotonated to COO-. Glutamate is the most abundant excitatory neurotransmitter in the brain, the precursor to GABA, and the molecule behind umami. Arginine, guanidinium group, charge delocalized across 3 nitrogens, pKa around 12.5, precursor to nitric oxide. Lysine, terminal amino group, heavily modified on histones, essential amino acid. Histidine, imidazole ring, pKa of about 6, partially protonated at physiological pH, ideal for enzyme active sites and for oxygen binding in hemoglobin. 2 amino acids with 2 chiral centers, isoleucine and threonine, and 2 sulfur-containing amino acids, methionine and cysteine. Methionine sulfur is inert, cysteine is reactive. 1 achiral amino acid, glycine. 1 L but R amino acid, cysteine. 1 start codon amino acid, methionine. 1 helix breaker, proline. 20 amino acids. That's the set. Thanks for listening. This has been MCAT Notes to Fall Asleep To, Volume 1, Episode 1. Make sure to like, comment, and subscribe. Excited to see you in Episode 2.